Alright guys, so in today's flip video, we are actually going to be reviewing a couple of organelles and their functions that you should have learned in 7th grade, as well as adding the new organelles and their functions that we're going to need for this PBL. So we're going to talk about the cellular structure, function, and transport. So when we talk about cells, we know that there are two domains, eukaryotic and prokaryotic. And eukaryotic means that they have a true nucleus, where prokaryotic means they do not. And in the eukaryotic, there are lots of different types, and the two that you also focused on in 7th grade were animal cells and plant cells. So those are the ones we're going to focus on today. So let's start with an animal cell. This is a picture of an animal cell, and we are going to actually go through all of the organelles listed here, as well as their functions. So let's start with the cell membrane. Animal cells do not have a cell wall, and so the cell membrane is going to be that barrier. Then there's also the cytoplasm, the nucleus, the nucleolus, and so that's what we're going to go with first. So let's talk about what the nucleus is. You learned that the nucleus is called like the control center or the brain, meaning that it controls all the functions within the cell. One of its main functions is also to hold DNA or to store the genetic material. This is where chromosomes can be found. The nucleolus is actually the dark part in the middle of the nucleus. This is just another layer. And this part actually has its own function, and it makes ribosomes, which should be a new organelle to you. So like we said, the nucleus is the control center. It holds and stores all the DNA, and the nucleolus, which is inside of it, makes ribosomes. And we're going to figure out why that's important in just a second. So here's the nucleus, right? This whole thing right here. There are actually little holes. We call those nuclear pores. This is where things that are transporting things into the nucleus or out, like ribosomes, can move through the nucleus and nucleolus. So when ribosomes are made in the nucleolus and they need to be distributed throughout the entire cell, they can move through these nuclear pores. And so these little dots right here, these little pink ones that are moving all throughout it, those are the ribosomes. So let's talk about, what a ribosome is. A ribosome's whole entire function is to make proteins. Now that we've talked about the 4 macromolecules through biochemistry, we know how important proteins are, not only to cells, but to all life-sustaining functions, right? Enzymes, they store energy, they have many, many functions. All hormones are proteins, so on and so forth. So ribosomes make these. They are actually also found in all types of cells because all cells need proteins, whether they are the most complex eukaryotic cells to the most primitive prokaryotic cells. Let's talk about the endoplasmic reticulum, which is abbreviated ER. Their nickname are called the roads, and their function is they are the internal delivery system of all cells. So when those ribosomes are created in the nucleolus and they come out, the endoplasmic reticulum is a way for those ribosomes to move throughout the cell to get to the locations that they need, as well as the proteins that they make. So this is why we call them the roads. You could also kind of relate it if you wanted to create an analogy to the circulatory system of the human body. How we talked about the circulatory system's main job is to deliver things. The endoplasmic reticulum isn't necessarily its job to deliver, but to provide a structure for things to move around on. So, go through this again. There are 2 actual types of endoplasmic reticulum, and one is called the rough ER because it actually has all these little bumps on it, and it makes it look rough. You'll notice the rough ER is always going to be right up against the nucleus. The other type is called the smooth ER. It's the exact same thing, however, it doesn't have all these little bumps on it, hence the name smooth. So let's talk about the 2 types. The reason why we call the rough ER rough is because it has ribosomes on it. And since it has ribosomes, you guessed it, its function is to make proteins. So the ribosomes come out of the nucleolus and they attach, some of them do, to the endoplasmic reticulum right outside, while others are freestanding and they move throughout the cell. So their job is to make proteins, because like we said, proteins are so important, you can't have enough things making them. The smooth ER, however, is called smooth because it does not have any ribosomes, so therefore it doesn't make proteins, but it makes another macromolecule that is just as important, and that is lipids. So we're going to actually go into very detail of the cellular membrane, but if you haven't kind of already noticed from your word wall, we know that a type of a lipid is that phospholipid, right, that has two fatty acids and a phosphate head. And we know the phosphate head is hydrophilic and it loves water, and the fatty acids are hydrophobic and they hate water. So to protect those fatty acid tails, they make what we call a bilipid layer, and that is what a cellular membrane is. So rough ER, smooth ER. So let's talk about the Golgi complex. The Golgi complex is nicknamed the shippers. You can think of them as like the UPS or the FedEx of the cell. Their job is to package, modify, and transport things throughout the cell or even outside the cell. So if water is coming in or glucose is coming in, it decides where it needs to go. If carbon dioxide and all these waste are created as breaking down that glucose, it needs to transport them and package them so that they can be shipped out of the cells and picked up by the red blood cells to be removed from the body. So Golgi is the UPS. They package, they ship, they distribute. And they kind of look like a stack of pancakes that are on the side, and you can see them right here in the blue. Let's go through all that. So this is the Golgi bodies. And what happens is actually they kind of like pull off and then make little bubbles, and that's how they're packaged. And then they're sent on their little way. And so another one we're going to talk about is lysosomes. Lysosomes are circular, but they're not a ribosome. They're bigger than a ribosome. And we call these our cleanup crew because their function is actually to break down food into particles the rest of the cell can use, and also to destroy other parts that are not working efficiently or are damaged. So they have a bunch of digestive enzymes in it, and we know that enzymes, again, are proteins that are made from those ribosomes. And so they kind of go through and they break down the glucose into a form that the mitochondria can use. And I don't think it has lysosomes in this picture, but if there is one, it would be like a small little circle, like around there. So let's talk about the mitochondria. You know the mitochondria as the powerhouse. And the reason why it's called the powerhouse is because it makes energy from the cells. Well, it actually doesn't make energy because we know, according to the law of conservation of energy, that it cannot be created or destroyed. It is just transformed into a form that we can use. And the process is by breaking down that glucose sugar, which is a carbohydrate, which is a monosaccharide, into a form that all cells use for energy, which we call ATP. Now, this is probably the first time you've actually heard of ATP, but it's going to become extremely important and relevant throughout our course of biology. So we definitely need to know that all living things use energy in the form of ATP. And we'll talk about what that abbreviation stands for at a later time. So ATP is a major fuel source for all cells and their activities. So here's the mitochondria. It kind of looks like a bean. It always has this squiggle line in the middle, which is what we call the matrix. And this is where the glucose is actually broken down and transformed into that ATP form that the cell can use. And mitochondria. And so then the last one we're going to talk about in an animal eukaryotic cell are centrioles. Now, centrioles, their function is to aid in cellular division. The cell can go through two different types of division: mitosis, which we will be definitely discussing later in this PBL, and meiosis. Normally, they are found near the nucleus, but they can actually only be seen when a cell is about to start the dividing process of either mitosis or meiosis. And they are made up of what we call microtubules. So when we look at this picture, here's the cell. The centrioles are right here. This is what they look like. They're kind of like little hollowed straws, but if you wanted to break it down, it's actually made of a bunch of microtubules together. So this is where we're going to stop the video for today so it doesn't get too long. And tomorrow you'll have the second part where we will talk about the few organelles that we haven't mentioned in plant cells. So make sure that if you have not already, that you go back and rewind the video, that you have a really good picture of all the organelles, the function, and the type of cells that they're located. I know mostly we focused on animal cells, which is our, of course, eukaryotic, but then we did mention a few that are prokaryotic. So the function and the structure are what you need. See you tomorrow.